Hello students, welcome to the previous year question series of CUET 2025. In this video, we will be discussing the questions which came in last year 2025 paper in SCQP12 Food Science and Technology. I'm Devanshu Gupta, your mentor for Food Science and Technology, and let's begin with the first question. So the first question which came in exam was, during the process of ripening of fruits, the translation of pectic substance occurs in the following order, and the options which they are given in protopectin, pectin, pectinic acid and pectic acid. So if we follow the correct chemical sequence, first of all, always we have protopectin, which is an insoluble pectic substance which is found in unripe fruits and which is responsible for the firm texture. So whenever we have a ripe, or we can say unripe banana, and if we compare it to the ripe one, so the unripe one is very firm, so it is because of the protopectin. The next which we come is pectin. It is formed by the protopectinase action and it becomes soluble and the fruit softens. So during the ripening stages, when the fruit softens, so pectin is being formed. Then we have pectinic acid. Pectinic acid is produced when pectin methyl esterase removes the methyl groups from the pectin. And the last we have the pectic acid, which is formed by polygalacturonase hydrolysis, which leads to over ripening of the fruits. That is why whenever we see the bananas which are over ripened, they are very soft and mushy. So the correct sequence is the one which is given in the exam, A, B, C and D. But we can see that there is no option which matches to that. That is why this question was dropped in exam. What does that mean? That means that if the question which is given by the authority, that means NTA, the question which they give, if there is a error in the question or there is an error in the options, in that case people who have attended the question will be getting full marks for the question. If you have not attended the question, you will not get any marks. That is why I often advise the students that always do 2 to 3 extra questions in which you are at least doubtful. Don't leave the question. Okay, so I hope the answer for this question was clear. Let's go on the next question. Identify the correct sequence of byproduct formation during cocoa and chocolate manufacturing process. So when we were discussing about the chocolate manufacturing process, we found out that first we have the fruit which is subjected to cleaning and fermentation, and let's say the options which are given: cocoa powder, chocolate liquor, cocoa butter and nibs. So I think nibs are the one which are initially formed, because when we grind the cocoa beans which are roasted, then we get the cocoa nibs. When cocoa nibs are subjected to extreme level of grinding, we get the cocoa liquor, which contains powder plus cocoa butter. When we filter the cocoa liquor, so this is always the first point, then you get the cocoa liquor. When you subject the cocoa liquor to pressing so that the butter is separated, you will get the cocoa butter. So cocoa butter is the third option, which is formed when you hydraulically press the cocoa liquor. And the last is cocoa powder, which you get after you dry the cakes which are left from the filter press. They are subjected to drying and pulverization. In pulverization, extreme fine powder will be made from the cocoa cake, and that is called as cocoa powder. So this is your fourth. So answer in the correct sequence is nibs, then cocoa liquor, then cocoa butter and then cocoa powder. Let's see D, I think third option. So C is the right answer for this question. Next question, if we go on, is which of the following statements are correct with reference to composition of fats and oils? First option is the chain length and the degree of saturation control the properties that are found within the fatty acids and lipids. Yes, this is right. Fatty acids typically contain an even number of carbon atoms. Yes, this is also right. The saturated acids have higher melting points than unsaturated acids of corresponding size. Yes, this is also right. So all A, B and C are right. So fourth is the right option for this question. Coming to the next question, which we have is, it is in the match the following type question and it comes from bakery. So here you have given the cake defects and here you have been given the causes. So let's see. First is too light crust. When we were discussing about the faults and remedies in bakery products, we discussed that too light crust is because of too little sugar which is being used. So A goes for 3. It is because sugar promotes caramelization and mild browning in the cake, and that is why your cake, when they are cooked, they get the brownish color. If you have too dense texture, it is because of too much liquid, because the leavening agent which you have kept in the cake is not able to promote the leavening of the cake, and that makes the cake very dense, and it is because there is too much liquid in the cake. Then if we see the third option, too dark crust, it is because of too high baking temperature. If the oven is operated at a very high temperature, the heat will not enough time to penetrate, and it will cause the burning from outside. And crumbly texture is because of too much shortening. So A goes for 3, B goes for 4. Let's see which option is right. Yes, fourth is the right option for this question. Then before we proceed on to the next question, I have a very short announcement for all the people. So I think you all might be thinking as to how to prepare for this exam, which is almost 10 days left. So for those people who are still worried, who don't know how to prepare, who don't have the proper notes, who don't have the proper sources, we at AgriVision for You have planned a batch strategically for only you people, in which we have launched a crash course, in which we will be discussing about all the topics which are coming in your CUET exam, followed by sectional test, full length test, revision, mocks, and one-on-one discussions on the doubts. So for all the students who have not yet figured out how they will prepare for the CUET, it's the last chance. You can get enrolled in the CUET batch. The number which is given on the screen, you just need to ring a call on that number, and the person will be helping you to get on board with us. And if you are watching this video anywhere in the mid of 2026, that means you have not given the 2026 exam and you are preparing for 27. So you can join our regular batch, which starts from August onwards, in which we discuss each and every topic word by word. So August to March is the duration of the next batch, that is CUET 2027. If you are watching in mid of 2026, then you need to join the full batch. And if you are watching before 2026 exam, then you can join our crash course, which will help you to cover the CUET 2026 and 2027's SCQP12 exam. Coming on to the next question, which we have is the process of homogenization. The process of yogurt manufacture involves the following sequence, and the steps which are given is homogenization of milk, standardization of milk, pasteurization followed by cooling and addition of starter culture, and fermentation. So always remember, standardization will be the first step which is given in a dairy plant, because as soon as the milk is received, you never know what is the SNF of the milk, you never know what is the fat percentage and all. So you will determine it via the machines which are available, and then you will add or remove the substances. For example, fat is extra, so you will remove some fat and bring it down to the required level. So standardization is always the first step. Then it is followed by homogenization, because the fat you have added or the fat you have removed, you need to standardize the fat in the whole mixture, so you will subject it to homogenization. Then you will subject it to pasteurization, so you need to decrease the microbial load before you are going for fermentation, as to promote the growth of only beneficial microbes in your media. Then you will be going for fermentation. So I think B, A, C and D, right? B, A, C and D, third is the right option for this question. Then coming to the next, increase in volume of bread during baking is known as, and this option is very clear because we have already discussed a lot about the bakery topic. This is known as oven spring. Now, why oven spring happens? Oven spring basically happens because of gas expansion, yeast activity, because yeast produces CO2, and the steam formation in the dough, because the dough also contains water. When it is subjected to such a high temperature, the water will be converted into steam, and that also causes the bread to leaven up. When the dough will enter into the oven, CO2 expansion will be the first step. Then it is followed by the yeast activity, which will cause the CO2 bubbles to also expand, and then water will convert into steam. So this is the correct sequence. Also, if you get a question in exam, that what is the correct sequence of the events happening in the oven, so this is the correct sequence, and oven spring is the right answer for this question. Let's proceed on to the next question. Next question is, during processing, the characteristic flavor developed in cocoa is. So this is basically coming from your Maillard reaction, and we have studied that in cocoa, coffee, the flavor is developed during roasting process. That is why whenever we go for best quality coffee beans, we always keep an eye onto for how much time they have roasted and who are the roasters, because everyone who is roasting is not an expert, right? So Roasting is the right answer for this question because roasting happens at somewhere 120 to 150 degrees Celsius and it triggers the Maillard reaction, causes the browning, causes the development of the nutty flavor and the aromatic compounds. Okay, and it also reduces the bitterness and acidity in the coffee beans, and these reactions generate the volatile flavor compounds which are responsible for the characteristic flavor and aroma found in chocolate. Coming to the next question. The enzyme used in isomerization of cornstarch during the making of high fructose corn syrup is. So options which are given is alpha amylase, glucoamylase and glucose isomerase. These all three enzymes are used in the isomerization. Let's see what is the function of each of them. Alpha amylase causes liquefaction. That means it breaks the alpha 1 4 glycosidic bonds and causes the mixture to liquefy. Glucoamylase causes saccharification, which hydrolyzes alpha 1 4 and alpha 1 6 glycosidic bonds. And glucose isomerase causes isomerization, which converts the glucose to fructose. And this is how your high fructose corn syrup is formed. And basically your HFCS contains around 42% to 55% of fructose. It is a very common ingredient often used in your beverage industries, your confectionery industries, because it will not crystallize and it is a very easy product. Instead of sugar, people sometimes use HFCS if they want the end product to be in the semi-liquid or liquid form. So the most appropriate answer is A, B and C. That is the fourth option. Let's proceed on to the next question. A flexible pouch capable of withstanding temperatures of about 120 degrees Celsius is known as. So I think many of you have seen some pouches like this in your surroundings, right? Often the ketchup pouches which are available from Kisan and many other food items are available in these pouches. These are called as your retort pouches because they contain multi-layer. They contain PET layer, then aluminium foil, and then polypropylene layer. Ideally these are in several laminations and that is why it is also called as a laminated pouch. Every layer has its own advantage. PET gives heat resistance and strength. If you talk about foil, aluminium is absolute barrier. That means no gas, no water, no microbe can permeate through foil, which is aluminium foil. And then you have polypropylene. It provides a heat sealable layer. So you want inside to be heat sealable. Otherwise, how will you seal foil? If you press two pieces of aluminium together and you apply heat, they will never join. But when you do the same for polypropylene, they will join because it will melt at a somewhat higher temperature. So this pouch is used for storing sterile food items. That means the food items are stored in it and then it is subjected to a very high temperature, usually the same treatment which is given to canning, and then the food which is inside it is completely sterile, and sterilization is done at around 121 degrees Celsius, which is almost near to 120 degrees Celsius. So third is the right option for this question. Then let's go to the next question. The yellow color of the cow milk is due to the presence of, and we have discussed this n number of times in the batch, that buffalo milk is white because of casein and cow milk is yellowish because of carotenoids. So fourth is the right option for this question. Now, how this actually happens? Cow convert the dietary plant pigment. That means whatever the fodder they eat, they convert that plant pigment to partially beta carotene. Now beta carotene remains in the milk fat, which gives the creamish yellow color to the milk. So if on one hand you have cow milk, on one hand you have buffalo milk, just by visual examination you can tell which is cow milk and which is buffalo milk. Buffalo milk is usually white because even buffalo eats the same fodder, but it converts the carotene into vitamin A. Now if you have seen the structure of carotene, you will find that it is almost like this, and when it breaks from between, it produces two molecules of vitamin A. So buffaloes convert the beta carotene into vitamin A. That is why the milk is not having the yellowish color, it is having the whitish color. But cows do not convert it. That is why cow milk is slightly yellowish in color. So buffalo milk appears whiter because of high amount of casein, followed by the conversion of beta carotene into vitamin A. But cow milk is yellowish because it contains beta carotene and it contains a lesser amount of casein. Then, if any students who is watching this video has any doubt in any of the option or in the entire syllabus of CUET, they can drop their questions in the comments or they can contact on the number given on the screen. We will be very happy to clarify any of your doubt regarding any exam of food. Not even CUET, if you are preparing for NIN, if you are preparing for GATE, if you are preparing for CFTRI, if you have any question which is you are not able to encounter during your preparation, you can always contact us and we'll be very happy to clarify any of your doubt. Then coming to the next question, QRAC stands for. So this is a very common question coming from your food safety or let's say food quality management. The answer is Quantitative Risk Assessment Committee. Now what is the role of this committee? This is a committee which deals with food safety and risk assessment. Risk assessment, risk management, and risk communications are the major roles of this committee, and it performs their task to identify the exposure level of the chemicals, to identify the probability of any adverse effect of any activity, and to identify the risk of that activity to the population. So these are the functions of this quantitative risk assessment committee. Let's go to the next question. Arrange the following steps of jelly marmalade processing in a proper sequence. The options are selection and preparation of fruit, preparation of shreds and peels, boiling and cleaning of pectin, and canning and sterilization. So if we actually see, they both all options are arranged in the correct order. That means A, B, C, and D. So let's see one by one all the options. In selection and preparation of fruit, you will take the fruit, you will clean it, you will make it ready for the next step. Preparation of shreds and peels, you will cut the peels into thin shreds so that they are easy to mix in the entire mixture. Then boiling and cleaning of pectin, peels will be boiled with sugar and acid, pectin will be extracted from the peels, and the foam and other impurities will be released from the mixture, and then you will fill it into the proper containers, either your cans or your glass containers, and then you will be giving the heat treatment for the shelf stability. So A, B, C, and D. Let's see which option is right. Second is the right answer for this question. Coming to the next question, the molten mixture of sand, soda, limestone for the development of glass is called as. So basically, when we manufacture glass, we make a mixture, molten mixture of all of them, and we fed that mixture into containers so that it is formed into a particular shape, and that is called as gob. Always remember this mixture, molten mixture is called as gob. Basically, three raw materials are there: sand, soda, and limestone, and they are melted to a temperature of about 1400 to 1600 degree Celsius, so that all of them are converted into molten form. Next question which we have is, match the following into the correct order of their storage temperatures. So banana is usually stored at 13 to 15 degrees Celsius to prevent chill injury. Then lemon is stored at 12 to 14 degrees Celsius so that it is also chill. It is also subjected to chill injury, but the temperature is slightly lesser for the storage than banana. If you talk about nuts, nuts are stored at a very low temperature of your 0 to 2.2 because they have high oil content, which can be subjected to rancidity, and to prevent their oxidative stability, we are storing them at a low temperature. And tomatoes are stored at 4.4 to 10 degrees Celsius because if you store at above 5 degrees Celsius, they will be subjected to accelerated respiration, which will eventually cause them to ripen over time. So A goes for 1, B goes for 3, C goes for 2, and D goes for 4. So I think second is the right option for this question as per the options we have matched. Coming to the last question of this video. The instrument which is used to measure the salt percentage of pickles is known as, and the people who are in the class, we have discussed this enough number of time that the option is salt meter. Salt meter is the instrument which is used to measure the specific percentage of salt in the pickle. If you talk about polarimeter, it measures the optical rotation of sugars. If you talk about the brinograph, it measures the dough rheology in bakery. And if you talk about ionograph, it is used in ion analysis. So salt meter is the right option for this question. And here we come to the end of this video. In this video, we will discuss the 15 questions which came in CUET PG 2025. You can follow us on all the social media handles to get regular updates. We keep on posting the information which might be helpful for you. Let's say in any of the agricultural based exams, food tech exams, any of the admissions, even job postings. This was all for this video. Stay connected with us. We'll be soon posting the part 2 of this video, in which we'll be discussing the further 15 questions which were asked in CUET PG 2025. Let's prepare best for the 2026 exam. I was Devanshu Gupta, your mentor for food science and technology. Thank you for watching.